Our next speaker is Uri Weill from the Weizmann Institute.
Hello, everyone. I want to thank the organizers as well. My name is Uri Weill from my Schuldner's lab, and today I will be telling you about a new technology for the characterization of the yeast proteome. So, at our lab, we are detectives of the cell. We want to know what is happening in the cell and in the proteins that compose it. To date, about 50% of the yeast proteome proteins don't have an assigned localization or function in the cellular landscape. Now, a cellular landscape is somewhat similar to the landscape of the city. And when you want to know what different people in the city are doing, a good place to look would be to ask where they are located. So if we know this person here is located at a hospital, we can assume she's a doctor or a nurse. And so the same is true for proteins in the cell. When you want to know what they're doing, a good place to start would be to ask where in the cell they're located. A very strong tool to investigate protein localization is to tag proteins with a GFP, a green fluorescent protein. In Saccharomyces cerevisiae, this was done, and a genome-wide collection was made in which each protein was tagged at its C-terminal with a GFP. This collection has facilitated the characterization of many proteins, and over the last decade, many studies have been carried out amounting to amazing insight into the yeast proteome. Despite that, to date, about 2,000 yeast proteins have not been accurately imaged or localized with the GFP C-terminal collection. This is because of two main reasons. First, the C-terminal tag may interfere with C-terminal motifs required for localization or stabilization of the tagged proteins. And so GUS1, which needs to go to the cell wall, is mislocalized. Second, the native abundance of many proteins is just too low to be imaged, so you can't really see where MEM2 goes to. A solution to both of these problems can be achieved by tagging proteins at their N-terminus. First, an N-terminus tag does not interfere with C-terminus motifs. And so GUS1 gets to the cell wall. And second, the N-terminus tagging cassette includes a generic constitutive promoter that gives high expression of the tagged protein, and so we can see where MEM2 goes to now. And so the aim of our project was to construct an N-terminus GFP tagged yeast collection. But creating a yeast collection is laborious, time-consuming, and very expensive process. And this is why so few genome-wide libraries were ever made. And so, in addition to complement the already existing C-terminal GFP library, we wanted our library to serve as the basis for the creation of many other libraries. To that end, we have collaborated with Anton Kamelinsky from Michael Knupneb, and that has developed a novel tag swap strategy. In this strategy, our basic GFP tagged yeast collection that we built can undergo swapping of the tag into any other desired tag. Now, this swapping process is easy, fast, and relatively cheap, yet it has also been proven to be very accurate and efficient, allowing anyone to create a new collection, genome-wide collection, in the time it takes to do an SGA, which is about 3 to 4 weeks. So for the application of this strategy in our project, we have chosen the name swap tag, or SWAT for short. Recently, we published the first step in this project, which included more than 1,400 proteins that localized to specific organelles. In addition, it included more than 300 proteins that bear a signal peptide, which were tagged with a specified cassette that included in it a generic signal peptide. Now, both of these libraries have the SWOT ability that I told you about. So once we constructed this library, the first thing we did is to image it with a fluorescent microscope. And so we set out to compare our localization data to that of the C-terminal GFP library. We found that hundreds of proteins could only be visualized with an N-terminus tag. In total, we found that more than 500 proteins that couldn't be seen with the C-terminal library are now seen, and for those we assigned new localizations. In addition, it was quite reassuring to find that more than 700 proteins have the same localization, whether they are N or C-terminally tagged. And finally, it was very interesting to find that more than 300 proteins have a different localization, whether they are N or C-terminally tagged. And it still remains to be seen which of the tags is the correct one. So many of these newly observed proteins have intracellular localization, which is quite easy to determine. I mean, if I see something like that, I know that it's pretty much sure to be ER localization, and the same thing is true for like mitochondrial localization. But for other intracellular localization, specifically punctate form localization, you need a specific cellular marker in order to determine localization. So you need to know if it's peroxisomes or lipid droplets, Golgi, many other possibilities. To that end, we have introduced several well-characterized markers, each tagged with a red fluorophore, into a subset of our library. And each one of them tagging a specific compartment that you can see here. And we imaged all the strains, and now we're doing the analysis for co-localization that will tell us if those protein resides in those compartments. Our analysis so far has helped us determine, indeed, localization of many proteins in those compartments and better define the protein roster within each and every one of those compartments. If anyone is specifically interested in any of those compartments, please come speak with me. I'd love to hear it. So just as an example, here is the analysis for the peroxisomal compartment, and we found that the majority of peroxisomal associated proteins could only be visualized with an N-terminus tag. In addition, this analysis has helped define 8 new proteins that colocalize with peroxisomes. So, okay. Now, in addition to inspecting protein localization, we also assign and determine protein abundance for the entire SWOT collection. Now, I remind you that the SWOT tag includes a generic constitutive promoter. Despite that, we found that protein abundance varied dramatically, indicating that there is massive post-transcriptional regulation in play here. So at that point, we wanted to utilize the SWOT ability that I told you about for the library. Now, a very important aspect of the SWOT library is that it is possible and easy to restore the native regulation of the native promoter of the tagged protein. You do that by excising out the tagging cassette and leaving in only a GFP. We call this seamless swapping. So to see if this works, we tag the well-characterized, low-phosphate-induced FO5 protein. And you can see that under the regulation of the generic promoter, FO5 is abundant in both high and low phosphate conditions. But after swapping to the regulation of its native promoter, FO5 is only abundant in low phosphate condition, telling us that seamless swapping can indeed restore the native regulation of the promoter. So once we saw this is working, we did that for the entire array. And now we have 2 libraries, what we call the generic GFP library and the native GFP library. And we imaged all of them, and we assigned protein, and we determined protein abundance, and we compared one to another. And, well, as expected, for many proteins in the native library, you just, again, can't see them because they're too lowly expressed, but then are well represented in the generic library. And that is why it's important to have both libraries that complement each other. In addition, we found a trend in which for many proteins, their native abundance correlated to their generic one. meaning that additional levels of regulation are in play here. And so what we did is we compared our data sets to already existing databases, and we found that while protein steady-state abundance does not correlate well to protein half-life, it does correlate to protein translation rate obtained by ribosome profiling. So we hope to further investigate these findings in future research. So both GFP libraries that I told you about are very strong new tools. But the very exciting aspect of the SWOT library is that you can actually change each and every one of the components to whatever you like. So you can change the selection, you can change the promoter, you can change the tag to give you endless combinations of creating many yeast libraries. To see if this worked, we indeed changed the selection, promoter, and we changed the tag fluorophore from a green one to a red one. And you can see here that where before you get a strong GFP signal, after swapping, it is abolished, and you get a red one instead, meaning the swap worked. And once we saw this is working, we did it for the entire array again, and now we have it in both green and red. And now it actually allows you to do colocalization assays where you take strains from the green library, take strains from the red library, cross them, and check for colocalization of the tagged proteins. So to finish, now that we are certain in the power of this technology, we are expanding the SWOT collection to include the entire 6,000 yeast proteins, including more than 400 proteins which were specifically tagged for that bear a mitochondrial targeting signal, a very important N-terminus motif. And finally, we are now constructing additional tagging modules, creating new libraries with new promoters, new tags, different components. And I'd love to hear from you guys if you have any ideas for anything that could be useful, any library that you would like, please come up and talk with me. And I would like to thank Maya, who's a great advisor, Ido, who's been leading this project from the beginning with me, Sylvia, Omer, the entire Schuldiner lab, our collaborators in the Knoop lab, Anton and Matthias, and all the collaborators. And again, all of you for listening to me. Thank you very much.
In the back, David?
For proteins that are supposed to go through ER and Golgi sorting, how with an N-terminal GFP are they gaining entry to the ER, or do you know that?
So, as I told you, are you referring to the single peptide that would be interfered with? So, as I told you, we tag those proteins specifically. Well, first we characterize them. That was not an easy task. And then we tag them specifically in the cutting site in the cleavage site with a cassette that includes a generic constitutive promoter that we tested several to see that it's the best one to insert it into the ER and get it. And indeed, when we look, we see that it does go into the secretion system.
And is that left behind when you cleave out and go back to the native promoter?
Exactly what you mean. So once you do the seamless swapping, you restore not only the native promoter, but also the native signal peptide.
So did you see any variation in the expression of ones that were put near like subtelomeric? Were there any silencing effects that you could see that were overcome by the strong promoter?
Oh, so actually we didn't check. That's a really good thing to look at, and I would check my data and see if there's anything interesting there.
To do the swap into both, so you can mate red and green, you have to have them in both mating types. Are you constructing them in that way?
So the green one is A and the red one is alpha. So that is what is available so far. Yep. But if you want it, I mean, it takes 3 weeks and you can have it in the other one as well.
It's Burger King. You can have it your way. Thank you very much.